Hi, welcome to the ISBT Tissue Culture room of 261 in LaSalle's new Holroyd building. It's beautiful. Welcome. Now, as soon as you first walk in, you'll notice that there's enough bench space for you to work on. You can come into this room, and this is a room where you do a lot of the prep work. And we also have microscopes here as well if you need to count cells. We do a lot of cell counting in here. It's a nice big space. And if you follow outside of the room, you'll see that there's the fridge and the freezer right here. This is where we keep a lot of the media to grow the cells. Nice and clean. Everything must stay clean and sterile. And the freezer as well. It's empty right now, of course. Again, more counter space. This is a water bath to melt any of the media out of the freezer if you need to. This is one of our rooms where we have our laminar flow hood where we do all of our work at. And also each room has 2 incubators. Sometimes they're set at different temperatures. There's one on the bottom that's set at 30 degrees Celsius and the one at the top is set at 37 degrees Celsius. Your cells grow at different temperatures. Here you can take a look at inside. They also have a HEPA filter in them as well. It's very similar, the same filter that is used in the laminar flow hoods.
Right here is our laminar flow hood, and you always have to use the aseptic technique before entering your hands into the flow hood. It's mandatory for cell culture or you'll always get contamination with bacteria. Right here, let me wash my hands. Right here we have cell culture flasks, just 2 different sizes. Here we have a filter to filter our media through. And here we have a pipette to pipette our media into our cell culture flasks. This, as Nicole said, this does have a HEPA filter. And that's pretty much it for the laminar flow hood. Here are some guidelines for aseptic technique before using the laminar flow hood in cell culture.
Always wear gloves and make sure you wash your gloves with 70% ethanol. And then before you do anything else, make sure you wash out the hood with 70% ethanol. Always wash your gloves first before entering the hood. Anytime you take your gloves or your hands outside the hood, you always have to rewash them with 70% ethanol. So when you put anything in the hood that you're using to prep for cell culture, you always have to make sure they're sterile. Even though they're in a sterile bag, you have to make sure the outside is sterile as well. So wash it with 70% ethanol. And then place it in the hood. Same as for pipettes, markers that you need to label. I got it. Oh, okay. Or any markers that you need to label any of your plates. The plates that are in there right now are already sterile, as well as the little centrifuge tubes. We sterilize them in the autoclave. These are the guidelines before using the hood. Here's the liquid nitrogen door. Before you open it up, you always have to check out to see what vial you're getting out of the logbook. It's labeled by location. Here are the vials. They're called Kairos. Here's an example of where you can find your vial. You just lift it up, and the Cairo tubes are labeled by number as well.
These are actually frozen cells that we worked with last year.
And they just snap out.
Yeah.
And you gently place them back in. Okay,
Make
here's
sure
our
you-
other cell culture room. Right next door to the other one. Very similar setup.
Right here we have a hot plate to defrost antibiotics or media that we need to. Right here we have a single incubator. Very similar to the one next door, it just doesn't have one on top of it. Here's a look at inside. This one you can also change the temperatures to what the cells demand. This is the newest laminar flow hood that we have in the Holroyd building. And here's the control panel to turn on the light, the blower. This also has a HEPA filter. And lastly, here is our phase contrast microscope, and we use this to look at our cells. That just about wraps up our tour. Please contact Nancy Jones, the chair of the ISBT department, or Marsha Timmerman if you would like to take a course in cell culture.